Hi guys, Dr. Orlando here with a video on cell cycle controls. We're going to talk about how the cell cycle is controlled by certain proteins. There are many of them. We're just going to focus on a couple that help us understand how we can stop the cell cycle if something is wrong, and what happens if those things go bad. So this is cell cycle controls and lack of control. All right, so first of all, let's talk about checkpoint proteins. So you guys have seen this picture before. This is the cell cycle. The blue here is mitosis. We go into G1, gap 1 for growth. We can do G0 if we're going to go out of the cell cycle and just go do our job. But if we're going to replicate and keep going through mitosis, then we have to go through synthesis, copy the DNA, and then we have to prepare for mitosis in gap 2 and continue on. All right, so all along the way there are these checkpoints to make sure that everything is okay. So there is a checkpoint here right from G1. Make this like a stop sign, okay? But it's round. And this checkpoint is there at G1 to make sure that the DNA is okay. So these are proteins that check and see, is the DNA damaged? Is the DNA okay? Is it right? Okay. Then we have other checkpoints. So we have a checkpoint here. Okay, right after synthesis. Whoa. After synthesis is done, and this checkpoint says, did we replicate the DNA correctly? Was the DNA copied correctly? And again, these are proteins that are checking, inspecting, like inspectors, if you will, the DNA. Okay? We have a checkpoint between G2 and mitosis. And at that one, for G2, there's a couple things that we need to check. A lot of things have happened here. For this one, we're checking to make sure, did we copy all the DNA? Okay. Was there any damage? Were damages repaired? Damages, mistakes. Okay. And finally, is the spindle ready? Okay, do we copy our centrioles? Do we have our machinery in place? And then last, this spot right here, let's make it a little bit bigger. If we were to zoom in on these layers here, okay, we would see that we had, remember this was prophase, metaphase, anaphase, telophase, and cytokinesis. Okay, and I should put this in the right spot. Excuse me. So between metaphase and anaphase, there's a checkpoint. And I'll put a little M on that one. That one says, are all the chromosomes where they should be? Are all the chromosomes attached? Are all the chromosomes on the equator? Okay, so it really is checking to see if the spindle's built and everybody is attached. Okay, so those are the places that proteins check, and if there is a problem with any one of these, then it's the protein's job to halt mitosis. So if we fail a checkpoint, then the cell cycle should stop. It should halt until it's fixed or the cell dies. Either one. Okay, until it's fixed or the cell dies. Now, we know that sometimes mistakes happen. So how does it occur? Well, let's think about that. I said these guys, these checkpoint proteins, they are proteins. Okay, let's go to the next page for this, just to understand this concept. The checkpoint proteins, they are proteins. Where do proteins come from? Where do they come from? Well, remember, DNA is used to make RNA, which is used to make proteins. Okay, so these proteins here are used to check the DNA. Can you imagine where this could go wrong? Certainly. If your DNA is damaged, if the DNA that codes for checkpoint proteins is damaged, the cell will be able to continue to replicate, and that will lead to not such good things. Could lead to cancer. Oh. Cancer. Okay. All right, well, let's look at how that can occur. So the checkpoint proteins that control the cell, as I said, they're proteins. They are enzymes. Okay, so let's have a look at one specific pathway here that helps control the cell cycle. And I've made my little key characters here. I'm going to label them all for you. So this first brown thing here, I'm going to write GF on it. This is our growth factor. This is the signal that tells the cell to divide. Okay. This is the growth factor receptor. Okay. So that's what the growth factor will land on. This green triangle is, I'm going to put a C on it for cyclin. Okay, we'll see what cyclin does in a second. This is, purple guy is CDK. Okay. CDK stands for cyclin dependent kinase. Okay. Dependent on cyclin. Okay. Little P thing here. This is our phosphate. RB, I'm showing it like a door. Okay, so RB is a protein. This is a protein that will allow replication to occur. That can allow replication to occur. Finally, we have our DNA molecule here. Okay, so we're going to put all these guys together into a little picture here, and this will be our key. Okay. Okay, so let's start building this pathway. This pathway is showing the role of CDKs, so the job of CDKs. Okay, so here we go. Let's start moving these guys over. So first we're going to put, this is my cell over here on the right, the yellow lines, that's its cell membrane. I'm going to put the receptor right there. There's my growth factor receptor. I'm going to take the growth factor. Whoop. And so let's put everybody in place. So the growth factor is going to go there. CDK is going to be in the cell. Okay. With the there's phosphates in there. Okay. So then RB is a protein. And RB, I'm going to put him here. Move it up a little bit. I'm going to put it in front of the DNA. Okay? Like a door that leads to the DNA. Okay? I just want you to think of it like a locked door in front of the DNA. So here's how it goes. The growth factor, which is a signal being sent to the cell, this could be a hormone, it could be protein, it could be coming from somewhere else in the body through the bloodstream, or it could be coming from the cell next door. Either way, it's a signal that came from a different cell, and it's telling this cell it's time to go through mitosis. It's time to copy yourself. So the growth factor is very specific, and it will dock here with the growth factor receptor. Okay, so it will be activated. When that happens, it will trigger the cell to make cyclin. Okay, so there's our cyclin. Okay, that up. And it'll make a bunch of cyclin. The cyclin is going to go to the cyclin-dependent kinase. And remember what kinases do. What do kinases do? Kinases transfer phosphates. So once CDK becomes activated, because it has a cyclin-dependent kinase. The kinase activity is dependent on this protein binding cyclin. Okay, that's what happens there. Then it'll take the phosphate, let's grab him, and it'll bring it down here to RB, which I said RB is like a locked door. Its job is to block DNA replication, okay, until CDK phosphorylates it. Okay. When CDK phosphorylates it, the door is open. And when the door is open, it can go through, and the DNA will be copied. Like that. So you can say that RB controls the cell cycle, that it is a checkpoint protein. Okay? It doesn't allow DNA to be copied unless it is phosphorylated. Okay, so now let's look at other ways that the DNA synthesis can be controlled. We can control the cell cycle. So there's a gene called P53. This is in the DNA, and this gene codes for proteins, and these proteins' job is to look for DNA damage. Okay, so I'm going to just kind of move things around here on the picture quickly. I'll be right back with you. Okay, so I brought a new guy on board here. This protein, I made him like a guy with glasses because he is going to look at the DNA. He's inspecting the DNA. And his name is P53. The gene is called P53 and it codes for proteins that inspect DNA. So here's my scenario. He's going to look at the DNA. And let's zoom in. Look what I did to my DNA. I damaged it. You get a big chunk missing out of that DNA. Okay, so I made him kind of unhappy in this picture here. He's not pleased with the state of this DNA. This is already happening here. The growth factor has come on board. It's bound to the receptor. The cyclin's been produced. The cyclin is attached to the CDK, and the CDK wants to bring this phosphate down here and phosphorylate RB. Okay. Well, guess what's going to happen? I'm going to move him over a bit. The P53 proteins are going to say, no, this DNA is not okay. And they will come over here and they will block CDK. Okay, so now CDK can't get over there. He's going to try to get to the RB to phosphorylate it, but it can't because P53 says that the DNA is bad and we're not allowed to copy it. If you fix your DNA, then you'll be allowed to copy it. But at this point, we're shutting down the copying. So there's, whoop, draw. There'll be no copying. You cannot get in. So that's the job of P53. Okay, block CDK until the DNA is fixed. So we could kind of see where things could go wrong here, right? We can see where we could have a problem with this. So here's how it can go wrong. And before I get to there, I just have to do a definition to make sure you understand these terms. The term Protooncogene. When you hear Protooncogene, think of these are the good genes. These are genes that code for regulatory proteins, that code for the checkpoint proteins. So these are genes that code for proteins that promote mitosis. So like I said, these growth factor receptor, cyclin-dependent kinase, RB, those are all proteins. Well, RB doesn't promote it. These are proteins. We'll erase him. These are proteins that, and the growth factor as well, that promote mitosis. They promote it. So these are made from proto-oncogenes. The DNA, if you can find that strip of DNA and say this is what we copied to make this growth factor, this is the recipe that we use, we'd say that that recipe is a proto-oncogene. Okay, and a tumor suppressor gene is a gene that prevents unregulated mitosis. Unregulated mitosis. So we said P53 was the example here. I didn't draw him in my picture yet. We said that P53 is the one who can prevent unregulated mitosis. It's his job to say, hey, you guys are not checking your DNA well. You got a mistake here. We got to stop. So that's a tumor suppressor gene. A gene that, just to make the wording the same, gene that codes for, let me fix this. To a gene that codes for proteins that prevent unregulated mitosis. All right, like p53. Okay, so now we can go on and show you how this can happen. Okay, so we have this can go wrong by having DNA that is wrong, and we have our proto-oncogenes being mutated into oncogenes. An oncogene is a gene that is the mutated version of the proto-oncogene, and it's going to promote cancer. We could also have mutated tumor suppressor genes. Okay, so when we look at our picture over here, the oncogenes that we are going to look at being mutated are going to be the GF receptor, cyclin, and CDK. Okay, so these guys are the oncogenes, or will be oncogenes if we mutate them. And then the mutated tumor suppressors are going to be the RB mutation and P53 mutation. Okay, because both of those do suppress mitosis, right? P53 and RB are doors that keep things from happening unless it's right. All right, so let's go and look at these and make a little story about them here. So let's look at the oncogenes first. So just imagine, if I have a GF receptor that is turned on all the time, if it doesn't require the growth factor anymore, it's just on, it's going to tell cyclin to be synthesized at all times. It's going to have this CDK complex is going to be ready to do its job all the time. And we're going to follow this pathway of activation and copy DNA over and over. So it's going to have unregulated, uncontrolled DNA replication. Okay, and it's copying bad DNA, right? I mean, if we've got a bad protein up here that's turned on all the time when it's not supposed to be, where did that protein come? That bad protein came from bad DNA. So we're copying bad DNA over and over. So it could be the growth factor receptor that's mutated. We could have the cyclin being mutated. We could have the CDK being mutated. Okay, so those are all possibilities that would lead to uncontrolled DNA replication, okay? Just happening as it pleases. Okay? So those are examples of oncogenes. Now remember, tumor suppressor genes are good guys. They stop mitosis unless it is supposed to happen. So RB is an example of a tumor suppressor, is a protein that comes from a tumor suppressor gene, because it stops mitosis unless the correct pathway has occurred. And P53 is a tumor suppressor because he doesn't allow copying of bad DNA. All right, so these guys are tumor suppressors. Well, what happens if we mutate them? What if I make it so that the RB protein is always unlocked? If it's always unlocked, we're in trouble, because now we're going to copy our DNA over and over, and we're going to get, again, uncontrolled DNA replication. So all of the examples so far have resulted in an increased number of cells, and the cells themselves have, at the very least, have bad DNA that tells them to not control, that keeps them from controlling the cell cycle. At the very least, that's what they're copying. Okay? So then, let's look at what P53, what happens with him. Okay, so I damaged my DNA. Remember, P53's job is to find damaged DNA. And normally, he would go in there and he would block the door. He's a deadbolt on the door so that CDK can't phosphorylate it and we won't copy DNA. But now, P53 is looking the other way. He's mutated. Tumor suppressor gene has been mutated, so he doesn't work properly. If he doesn't work properly, he can't get in here to block phosphorylation, and the next thing you know, we're copying bad DNA. There you go. Okay, so now we've copied, replicated, damaged DNA. Okay. So you can see how these things probably go hand in hand. If you've got damaged DNA that affects P53, and maybe it affects also the growth factor receptor or cyclin or CDK, you're going to end up with something growing, a lot of cells growing, and they have bad DNA. So a big clump of cells with bad DNA means cancer. Okay. So that's how it can go wrong. We have to remember that the good guys are the proto-oncogenes, that they promote mitosis, and tumor suppressor genes prevent mitosis, unregulated mitosis, and that mutations can cause changes in those so that we end up with oncogenes, things that are turned on when they shouldn't be, and mutated tumor suppressor genes, ones that can't do their job. The door is not locked, RB is not a locked door anymore, and P53 can't stop replication of bad DNA, and you end up making copies of cells that have bad DNA, and that is what cancer is. All right, so that should do it for talking about the cell cycle controls and lack thereof. If you have any questions, please feel free to contact me.